Thanks so much for having me and for the kind introduction. Thank you all for coming. If you're at the back, there are still some seats at the front if you want to. So, Nita, I'd like to talk about recent and ongoing work in the lab on trying to extract mostly disease mechanisms from omics data, and with an emphasis on how we can leverage existing knowledge to do so. And to motivate that, I think it's helpful to think of omics data related to other data modalities where we have made great advances recently using AI in the broad sense of from deep learning algorithm to other modalities. And this is to look at this data according to, for a particular modality, how many observations we have and how many features we have. So we know the large language models. We heard earlier today from James, amazing work they're doing, how powerful they are for many tasks. But there we have a huge number of observations, essentially everything out there, and all these features, which are all the tokens. Another area where deep learning has been very powerful, which is image analysis. It's not as extreme, but also large numbers of both features and samples. And getting closer to the molecular domain in genetics with some of these large cohorts and the ability to look at all possible variants, we do have also relatively large years of observations and features. And for example, we heard yesterday in the dream session from Anshul Kundaji and others, great work in this space. Now, when we go to omics data, as we have moved from more classic biochemical technologies to omics, we have managed to increase the number of features, so we can measure thousands of proteins or metabolites or so forth. But we are not really able to do this as of now at a huge number of samples, let's say different patients. If we think of single cell and spatial technologies, and if we are able to agree that every individual cell is an independent sample, we of course increase the number of observations. But we also know that that's not quite the case, right? So single cells in a particular sample are highly correlated, so it's not quite the same. So we're still in a place where often we face a misbalance between the number of features and observations to have true statistical power. That's an important problem, and something which is also quite relevant in our view is that from just omics data alone, it's quite hard to get to true molecular causality. So to illustrate that, you can see here on the left a very well-known signaling pathway, the EF signaling pathway, which is targeted by many drugs, and it's very well understood, and there is a lot of knowledge, so we are able to draw the interactions between the different components. If we were now trying to reconstruct those interactions, those arrows, by looking at omics measurements, let's say the level of activity of some of its components, we will not be able to find the right topology because there will be many different networks that will be compatible with the data at hand. That's what you can see here on the bottom right. Now, if we're able to intervene, if we're able to block specific components, we can get much closer to true causality, and that's what this second set of figures tries to illustrate, and certainly that's a great area where there is a lot of development. But even then it's hard, and even if we have, even for the cases that we can do interventions, but also in many cases we cannot do interventions. Now, if we think of omics data derived from patients, we cannot just, of course, do different alterations on human beings. So that's kind of the motivation of why in our group we have this emphasis in leveraging prior knowledge in doing this type of analysis of omics data. And I will show you this is things that we get from databases such as which ligands binds to which receptor or which targets of transcription factor exist, to be able to reduce the dimensionality of the data and increase statistical power. And because we use this knowledge which is connected to biochemically well understood molecular mechanisms, we can get a bit closer to the causality. And as mentioned before, introduction, so everything I'll show you is provided as free open source resources that we're really happy if you use them. We have a very strong interest in benchmarking, almost, I would say, an obsession. And I will show you a couple of examples of how we do benchmarking in the lab, but we also do so in a crowdsourcing manner in the Dream Challenge that was mentioned before, and we had a great session about this yesterday. And then we apply this in our lab across different diseases. We are relatively agnostic in the application and spans from oncology, nephrology, cardiology, and autoimmune diseases. So if we want to use knowledge, we need to have access to it. And I just mentioned how powerful are large language models. So we could think of just going out there and ask a state-of-the-art large language model to build the knowledge for us. No, but if we were to go and ask GPT-4 to draw for us the EF pathway, it will give you a network that is nonsense. And this is something that is well known in the field, all these hallucinations, and there is a lot of efforts to try to overcome this. And so in that context, we built a framework called BioChatter, led by Sebastian Loventanzer, who is also in this meeting. It's a platform for, in an open source manner, to do this type of large language model-based applications. And the key idea is to ground it in the things that we know, that we trust, for which we use knowledge graphs in a framework called BioCipher. And of course, we are by no means the only one thinking of this. There's a lot of great activities. And again, if you think of what James talked about earlier today, it was fantastic work. So it's a lot of potential on these methods and tools. But as of today, at least, we are not at a place where we can fully rely on them to build the knowledge that we want to use to do science. And that's why what we use is manually, painfully curated knowledge available in databases. And what we built is a framework called OmniPath, which is just a metadatabase which has access to over 100 different resources about knowledge of molecular interactions, which are the components of a pathway, which ligands binds to a receptor, where are proteins expressed, what are the modifications in a protein, and with a strong emphasis in curation of knowledge. And the reason to have many databases, also sometimes databases that cover a similar domain, let's say pathway databases, is because different databases have different emphasis. Some may be focused on immune system, relevant for this session. Some may be more in oncology. Some will put a lot of effort in high quality, but then they will be sparser. Some are more lenient. And so in the same way that if you wear glasses, you may try a different lens to find the right lens for you. Through OmniPath, we do some benchmark, but also you can do your own benchmarks to find the right resource or combination of resources to do a particular type of analysis. Then we use this knowledge to, sorry, and I didn't know if it was working to work. Let me see if I can play this. I cannot. That's not so important. I only wanted to show you that actually we can use language models not only to generate the knowledge, but also to interact with the knowledge. And this is meant to be an agent that you can use to ask questions about specific molecules, and it will parse for you the knowledge to extract things of interest for your analysis. Anyway, so when we have this knowledge, we can use it to extract, as I said earlier, mechanistic features from omics data. And the type of feature you can extract depends on the omics data and which knowledge you inject. If you have transcriptomics, we can use it to estimate the activity of pathways by looking at the changes in the target genes of the pathways, which is what we get from the knowledge, which genes are downstream of a given pathway, or the activity of transcription factors in a similar manner. And we've done many benchmarks to see how to best do this, and then also apply the same concept to other data modalities. If you do, let's say, mass spec phosphoproteomics, you can look at changes in phosphorylation of proteins as a way to estimate the activity of kinases, which are the targets of, or the proteins that modify the phosphorylation of proteins, or you can use metabolomics to estimate the metabolic enzymes activities. And regardless of the omic data type and of the knowledge, at the end, what you do is some type of enrichment for which we built a package called decoupler. It's part of the ECCB core packages if you use that for single cell analysis, which allows you to perform in a fast and efficient manner many types of enrichments, and again, allow you to pick the right enrichment method for the question at hand. And these processes like transcription factor, kinesis, and so forth don't work in isolation. So what we also try to do then is to connect them and identify in large molecular networks what are the key paths that connect changes, let's say, upstream, triggered by a ligand in a receptor, through kinesis, all the way to transcription factor or to metabolic enzymes. And over the years, we have developed different methodologies and applied them in different contexts to try to do this type of analysis. Now, again, regardless of the data modality and of the question, from a pure methodological point of view, what you try to do in all cases is to input some type of knowledge, some signed or unsigned network, some type of omics measurement, and then use some algorithm, some method to identify those key pathways that are able to explain the data at hand. And these methods that can have very different nature. Normally, what they try to do is then to optimize a network to explain the data. And a couple of years back now, we tried to summarize a bit the existing methodologies. And some of you here have done great work also doing this type of analysis. This span from shortest path in a network to diffusion methods to all the way to neural or deep networks. And in our hands recently, we found two approaches to be particularly helpful. One is based on interlinear programming. This is a very efficient framework that allows us to find optimal solutions and often give us guarantee that we find the right model. And we develop a methodology to this type of analysis. But then in other times, it's more practical to use a more heuristic way. So we develop a propagation-based methods that allow us in a fast manner to find networks, and it's easier to recover the underlying algorithmic choices in how to find the pathway. So I will not talk much about the algorithms. I'm happy to talk more, but rather I will just illustrate it with one example. This is a relatively simple case study together with the lab of Raina Peppercorn, Amisha Savitsky at EMBL Heidelberg, where we wanted to study or find the key pathways that drive fibrosis in kidney. So we had a cell line that we induced with TGF-beta, a major driver of fibrosis. And at different times, as you can see at the bottom, we measure transcriptome, proteome, phosphoproteome, and in this case also the secreted proteins, the secretome. And then to link these processes to a downstream phenotype, which in fibrosis is the production of the cellular matrix. Using imaging technologies, we also measure those. And then generating this large data set, we kind of input into this type of methodologies I outlined before to identify which processes are altered in a TGF-beta dependent manner, but also try to understand how they are connected in a network. And one nice thing of this study was that time series that we have, so that we could not only look at a single snapshot of activation by TGF-beta, but try to have an approximation of how the network evolves dynamically after stimulation. So we could identify an early network of activation driven by TGF-beta stimulation, which in turn secrete proteins or other molecules that then gave rise to a second wave of activation. And you can see in the bottom two little hairballs that are not very, very helpful by themselves. It's just to illustrate a bit the size of the network. But in between, there is a heat map that tries to show that there is a major rewiring in the edges of this network from kind of the early times to the late times. And again, just to illustrate that it's a very dynamic process, how these cells are activated. But we think that the real value of this approach is then we can try to look into some more detail into these hairballs and look at the specific mechanisms that may be important in understanding, in this case, how fibrosis arise. And I want to emphasize that the way I think of these approaches is just as hypothesis generation tools, right? So we're taking data with all sorts of biases and limitations. We know our prior knowledge is also limited. The algorithms also have a lot of assumptions. So whatever we find in these networks, it may be true, but we always need to try to validate by orthogonal means. So an example of that in this data was that we found a potential pathway leading to extracellular matrix degradation downstream of TGF-beta, and this was mediated by the transcription factor E2F1 and the two genes you see there in green, SERPIN1 and 2. And again, this was an hypothesis, but to see if this is true, if this is a real causal mechanism, our collaborators perform a knockdown of E2F1 that then led not only to an impact on the cellular matrix production downstream there in blue, but also to the expression of the intermediate gene SERPIN1. No? And this is an example of how we can take a relatively complex data sets, multimodal, multi-time, kind of pass it through a network approach, identify potential mechanisms of interest, and then at least for one of them, lead experiment for validation. So I mentioned that there are many algorithms and there are many ways to do this type of network modeling. So for that reason, we are trying to bring together interested people, and this is also an open invitation from any of you working on this space to try to join forces and building an ecosystem of network-based approaches that we call network commons. So I saw you one example, one type of network, in our case from this Omnipath database, some types of omics data and one algorithm, but we can use different sources of knowledge, different omics data, and many different algorithms. So we have started to build such a framework, including some other groups like Evangelia Petzalaki, Livia Perfetto. But if any of you is interested in developing methods, but also, and in particular, frameworks to evaluate network models, and then we're really happy to talk more about this. And just as an example of why we think this is helpful, the idea of combining different algorithms of different nature. So we use network commons to combine linear programming, the framework I mentioned before, we develop with more downstream neural network approaches. And this is work in progress. But the idea is to just having one framework, but doing some type of plug and play of different methods, leverage, in this case, the strength of this linear programming framework called Cornetto that allow us to very efficiently optimize large networks across many different conditions to provide a kind of early scaffold to build a biologically inspired neural network that then generates a model that we can use for downstream tasks. So mostly what I showed you before was, I guess, implicitly talking about bulk data. And the example I showed you in fibrosis was all back technologies. But all of these ideas can be also applied to single cell and spatially resolved data, keeping in mind the potential challenges of, let's say, low gene coverage and so forth. And we have done this. But also, it allows us the opportunity to go beyond intracellular processes and to start to understand intercellular processes. And as you know, if you work with single cellular spatial data, there are many tools and great approaches that allow us to study cell-cell communication. And what most of these methods do is to say, if I have two groups of cells, two cell types or two cell states, and one of them expressing high levels a particular ligand, the other one expressing high level the cognate receptor, it is likely that these cells are communicating. And we're interested in this topic, and Daniel Dimitrov, at the time a student in the lab, developed a framework called Liana, which allow us in one place, again, to bring many of the existing tools, many of the existing databases for doing this type of analysis to study cell-cell communication. But here again, what we found particularly important to think is to benchmark. So how do we know if these methods work? Because if you think about the underlying biomolecular processes, and you think of a very simple context where we want to know if one cell A there on the left is communicating with cell B on the right, and what we have is single cell RNA, okay, we can see if the first cell is expressing the ligand, but then that ligand at RNA level needs to be translated into a protein, let's say a cytokine, needs to be secreted, needs to diffuse, needs to reach the target cell, interact with the receptor, and then kind of trigger a response. No, then is where we can really see there is cell-cell communication. But all we have is expression of RNA in both levels. We know expression on proteins don't fully correlate. There is a lot of processes in between. So Daniel, and in the context of the Liana development, but also in part in the open problems framework, and there is also similar work by other groups that show that these methods that are out there, they all can give you quite different results. It's not really clear which ones you should use. And most capture shock signal, but seems to be far from perfect. No? And we think this is because of the reasons I outlined before, like we are really looking indirectly at what's going on. And it's quite hard to have a proper ground truth for cell-cell communication. And I'm always happy to talk to anyone who has a good idea or a good system to check a place where there is true cell-cell communication in place. And we can really have ground truth. But from indirect benchmarks, we see that these methods work to some degree, but again, take this as a hypothesis generation tool and not something that you can necessarily take as a bona fide result. Another similar case is if we try to use single cell multimodal data to study gene regulatory networks. And as you probably know, we can estimate gene regulatory networks, use it to find the target genes of a transcription factor from single cell RNA. And if we have attack data, we have information about chromatin accessibility, we have, in principle, a much better chance to reconstruct the gene regulatory network. And then, in particular, after the development of publishing the attack method in 2015, But when Rostro et al., there was an explosion of methods to do gene regulatory network inference from single-cell multi-omic data. And then Pau Badia, who was a student in the lab, he's now postdoc with Anshul Kundaje, was interested in this, but then we realized there are so many methods that we thought rather than, again, a bit like in the cell-cell communication context, rather than trying to throw another horse in the race, let's try to take a step back and try to understand how these methods work, what they do differently, and maybe what are good practices for gene regulatory network. Now we have many methods. They all kind of use different strategies to go from single-cell RNA and attack to estimate the underlying gene regulatory network. And as an example of why this is important, if you take just a random data set, in our case from pituitary gland data, and you run several state-of-the-art methods that you see listed there, and actually at the bottom, the last one, Collect3 and Dorothea, are curated transcription factor targets, so it's not a GRN inference method, but just prior knowledge. We see that they give us quite different results, in particular at the level of the individual edges. No, the arrows in the network are quite different. So as in the cell-cell communication case, we also wonder which methods should be used. And also it's quite hard to have true ground truth for gene regulatory network inference. But then Pau developed a series of metrics to first compare and then evaluate the methods. And I will not go into the details, it's on the preprint, but it's not just which method does better for which task, but at the end of the day, all these methods have 4 steps. So from the calling to the pick map to linking the picks to the genes, and then Pau decompose all the methods in these 4 steps so we can do a bit of an analytical evaluation of the steps. And on the right here, we have gone into detail. You see some of the results. So many metrics, many methods. Some methods do better than others for a specific task. But in my view, this is not so much about which is the better method, but just how we can learn and find best practice and combine strengths of different approaches for certain tasks. So I'm now going back a bit more to the applications. So now in the context of single cell and single cell atlases, so how can we use these tools I just told you to, in the context of a disease, understand which programs, pathways, or gene regulatory networks are they regulating certain cell types, how we can connect this to potential changes in different cell states, and also, as I saw in this cell-cell communication framework, can we try to get a bit closer to understand how cell types coordinate in response to disease. And what we have found is a helpful way to look at this is to take existing atlases of public domain or with collaborators, take different samples. For each sample, try to do pseudobulk, so group different cells by a type or by a state. And at that level, try to identify major axes of variations that lead us to understand what's changing across a cohort of patients. For doing this, we use a relatively simple framework. So we repurpose multi-omic factor analysis, MOFA, from the lab of Wolfgang Huber, Oli Stegler, John Marioni, to find axis of variations which corresponds to multicellular programs across patients. There are many other ways of doing this, like dialogue, SCITD, many methods, or other methods more advanced than deep learning. But in our hands, at least, this simple framework is quite helpful because it's also very interpretable. And I will show you in a moment an example, but the idea is that we find these programs, these axes of variations that we try to use to understand at the molecular level what's going on, but also we can then try to link them to clinical descriptors or to map them to other data modalities from bulk to spatial data. So an example of that is immune-related and is kind of work in progress by Jose Linares Blanco in the lab. So Jose took 2 studies of lupus, so systemic lupus erythematosus is an autoimmune disease, and then reprocessed the data and then tried to find these programs. And the thing that we try to find is programs that include both shared effects. So for example, we would expect certain processes, if you have hypoxia, it will affect all the cells in a particular sample, but other cell type specific effects, maybe certain cells are more affected by inflammatory changes. As an example here on the right, you see one of these factors, one of these programs, which seems to be quite differently active in lupus patients versus healthy ones. and then how you can dip a bit into them and see which transcription factor activities are lower and higher across the different cell types. And if you can see on the right, a lot of the transcription factors that seem to be regulated are interferon mediated, so we can then try to go a bit deeper and understand what's happening. And we can do this by trying to build a multicellular network. Now, so what we do is to look closer into which cell-cell interaction processes are active in which cell types. You see here on the left, different cell types of kind of the level of interactions, on the right, which pairs of ligand and receptor seems to be more activated. But also, as I mentioned before, we use these programs as signatures that we can project in other data modalities. Oftentimes we have, for single cell, relatively small cohorts, but with bulk technologies, we may have larger cohorts. So here, Jose projected this factor, this program, on a cohort of longitudinal data for over 1,000 patients or visits of patients. And actually, this program, which was found in an unbiased manner, is able to nicely separate two types of patients, which is very important in lupus, those patients that have a long-term remission, so they're cured, versus those that have only a very short remission, and then the disease comes back in a few months. So this is just an example of how we used single-cell technologies or data sets with all of these approaches to extract disease mechanisms to understand what's happening and use it, in this case, hopefully to find ways to better prognose the diagnosis of patients. And so the last part, I wanted to briefly touch on spatial data. I think we are all aware that spatial data is great. It gives us a lot of extra context, very relevant for understanding many things from cell-cell communication to many other processes. In the lab, we have developed a number of methods. This was led by Jovan Tanevsky when he was a postdoc. Now he's a group leader in Heidelberg. And most of the things are published, so I will not go into details, but we have frameworks to decide spatial relationships in a global manner, to also do this in a more local manner using a variation of the algorithm, methods to combine single cell and spatial data. But because it's also in systems immunology, I wanted to say a few words of ongoing work to also in the spatial context try to combine gene programs with immune repertoire. And this is the work of Chang Lu, a postdoc in the lab, who's working with Camilla Emblom, who, when she was a postdoc with Joachim Lundeberg, developed a method called spatial VDG. So this is a technology where you can profile the immune repertoire in spatial resolution and combine it with spatial transcriptomics and vision. So what Chang is developing is a method that tries to kind of combine the spatial space with the immunological repertoire space and use it to kind of find similar and distinct features in the samples across these different domains. And as an example, then we can look at data. This is data from human tonsils. And the tonsils, there are different zones, the so-called dark zone and the light zone that play different roles in the maturation of immune cells. And we can try to start to understand how these variations in the clones corresponds to different locations in the space, and then which type of clones are colocalized, and then, of course, link it to what I showed you before of cell-cell communication, pathways, transcription factor, and so on. And then for that is where we go back to the cell-cell communication type of analysis I showed you before, where before I showed you in a single cell data, but of course, if we have spatial data, we can make a much more accurate estimate of cell-cell communication. We can look now at pairs of ligands and receptors that are nearby in the tissue, and they are more likely to be communicating. Now, if I'm close to you, it's more likely I'm talking to you that somebody at the back of the room can hear me, unless not with a microphone. So we can use that type of information to get more refined information about communication, and then try to identify putative mechanisms that drive cell-cell interaction in a space. As an example here, we revisit an earlier study of myocardial infarction, where we kind of knew we had macrophages and fibroblasts colocalized, and then using some of the tools I've been outlining today, identify a potential network that connects ligands in fibroblasts with downstream effects in immune cells, in macrophages, and seeing which pathways are triggered all the way to transcription factors. So with that, I'd like to finish and just share with you some of the things we're going to be working or we're working on now in the future. So we'll continue building prior knowledge resources. Now that we are, since a year back at TBI, we can really synergize with all the great databases there. We like to leverage knowledge graph and large language models, as I alluded earlier in my talk. We like to keep developing these models that combine intra and intercellular processes, combining knowledge and multi-omic and spatial data. But I think still is an important question in the field is how we go from what is until now mostly like static pictures on networks and diagrams that we can use to understand to true dynamic models that we can use to predict the effect of new perturbations in new readouts over space and time, but still leveraging the prior knowledge. I mentioned benchmarking. I mentioned earlier DREAM. And we had a great session yesterday. If you could not get in, watch the recording. We think it's really exciting times in computational biology where benchmarking is more important than ever, and we should come together to address this problem. And at the end, all this work, we do it for a purpose, which is to help to better understand human disease. And I'll show you just one key project for us that I think is very exciting and very immunologically related, which is that we started to work now in understanding the role of the microbiome in our gut, in our health. As you probably know, the microbiome, we carry all these billions of cells with very complex genomes in our gut, and we know it's involved in many diseases, from immune diseases like IBD to colon cancer. But the problem that we have is that we typically can look at is to look at the stool samples, which is far off from the gut. And as you see in that plot on the left, even the composition is quite different. So we like to find ways to look in situ at how the host and the microbiomes interact in our gut to better understand their effect on our health. So we started a project with Stefania Comelo, Georg Zeller, and Eduardo Villablanca, where we're going to build spatial atlases of healthy but also inflamed and gut and patients with cancer to map both the host and the microbiome tissue. And this is built on a technology developed by Stefania Comelo that allow us to look not only at the transcriptome of cells with spatial resolution, but combine it with a microbiome assessment using 16S technology. So this picture on the bottom left shows you the gut of a mouse, where you can see the lumen, the stool, the host cells, and how we can look at transcript levels, but also at the activity of or the presence of different microbiomes. So we like to build on this technology, combine it with perturbation experiments, what happens when you put microbiome, the host cells, and vice versa, and the mechanistic knowledge that I try to advocate for in my talk, to build not only multicellular, but actually multi-organism network models that combine the host and the microbiome, using to better understand how different microbiota lead to inflammation and other processes in our cells, but also ultimately to use it to find new biomarkers and new therapeutic opportunities. And with that, I'd like to finish thanking the people in the lab, our collaborators, our funders. If you like what we do, we're looking for postdocs in Cambridge. And I'll leave you kind of with the main theme of how we can use prior knowledge to help us analyze omics data. Everything I show you is free open source, so you can go there and download all the tools. And yeah, I'm happy to take any questions. Thank you for listening.
Do we have Bob Murphy or anyone for the second talk? I'm asking because of the, if not, then we will have lots of times for questions. I think we will have a little bit extra time for questions. So questions for Julio. Thanks a lot for the awesome presentation.
Hi, Julio, you know me, but maybe others don't know me. I'm Marta Milan from AstraZeneca, Oncology, Data Science, and AI. My question is on the benchmarking. So it's very interesting because, so after the talk, the keynotes that we did in industry, a lie costs a lot of money. In research, maybe, okay, a little bit of time lost in the lab in validation. So I was wondering, how do you see a collaboration between the industry and the academia to set standards of benchmarking that can help us both?
Yeah, no, I fully agree. And this is something we've been discussing also in other groups. And Gustavo is right there in front of you from DREAM. And I think so. And also because that's a space that is pre-competitive, right? So everybody would like to know which methods to use for what, and then you take it into your specific programs that you will be able to share. So to me or to us, I think it makes a lot of sense. And maybe building on strategies like open targets or other ways to come together in a pre-competitive manner, I think we should do that. Yeah.
So I also think that industry, some of the data is clearly private, but there is data that is there to share, could be quite helpful.
Yeah.
For benchmarking.
Exactly.
Clinical.
And there are also ways, even if there are data that cannot be shared, it can still be used for testing, right? So we also develop with people in DREAM, Gustavo, Justin, who maybe here, we develop ways to send the algorithms to the data and get just back the results. So even there are ways to work around the privacy for these purposes.
Question over.
Hi, Julio. Lovely talk, thank you. I'm curious, coming around to the cell-to-cell communication issues or the sort of accuracy of a lot of the methods in the field, and you talked about one major assumption being the fact that we're measuring mRNA and not protein. But I'm curious what your opinion is about the other assumptions that are being made with these methods, because even in a very simple controlled situation where you simulate ligands and receptors at single cell level, the performance is still, I'd say, below par.
I agree. And even I was surprised when we started to look at this. If you try to get estimates, let's say diffusion of molecules, we know really little, nothing. And of course, if the molecule doesn't go far, it will never work. And yeah, it's quite a complex problem, I think. And we think that we can get closer by combining different data modalities. So space helps. If you're closer, it's likely it's more signal, or if in the target cell, you think more of looking at the impact downstream as a proof of, if the ligand bound, it should activate this or that gene. But I agree, it's a very tough problem, and that's kind of the canonical cytokine mediated signaling. Many things are made by metabolites. It's very complex, yeah.
Maybe one last question, because our second speaker is here right now, so
Thank you very much for a very comprehensive presentation. There was a lot there that can actually make very different kind of researchers very, very excited. I was particularly interested in the prior knowledge work that you presented, and I was wondering what kind of prior knowledge do you think is most relevant that captures really the constraints imposed by biology? Is there a specific kind of data type or a specific kind of prior knowledge that perhaps is more useful? Does it mean that is it that one type fits all? Is there such a thing? But I was wondering what your thoughts on this were.
So that's a good question, and also depends on the question and the data at hand. So we typically start with the higher curated knowledge.
Can they use the laptop when you're answering questions?
Sorry, yes. So our study is often start with the more curated knowledge and see how far we can go. And sometimes you see that it's not enough, and then you need to inject knowledge that is maybe of lower quality or lower curation and go that way.